Hello and welcome everyone. I hope you had a wonderful day so far. And well, we are back here once again. It's another week of my IVF Answers webinars. So I'm very happy to be back with you all over again. But this time, of course, we do have new topics. This week, we will focus on embryology. So I'm sure you will have plenty of questions because this is a topic that I think that it's always great to be able to look what it's done in the laboratory actually. Yes, we don't know that much, I think, and it's really great that we are able to come here and find out some more. And we do have a very special guest with us tonight. This is Emily Baker. You can see her right here. Hi, Emily. How are you feeling?
Hi.
Thank you so much for joining us tonight.
No worries. Thank you for having me.
Thank you so much indeed. And I just want to mention that Emily is deputy laboratory manager at GENNET City Fertility, located in London. And Emily will discuss today what's happening in laboratories. She will explain the environment. And of course, she has a presentation on our topic. So yeah, we will start with that as always. But afterwards, remember, you will have a chance to type your questions in the chat section. Hello, Margie. Thank you for joining us tonight. And of course, don't forget, you can ask your questions during the presentation or afterwards, and Emily will be happy to help you out, no doubt here. She is the embryologist as well, and don't forget to ask her anything that is connected with the lab. She will be able to help, but of course, keep that in mind, please, of course. And yes, let's start with the presentation. Emily, are you ready?
Yeah, ready when you are.
Well, thank you so much indeed. And here's your presentation. Let's go ahead then.
All right, thank you. Thanks for that introduction. So yeah, I'm Emily. I'm going to be giving you a bit of a virtual tour around the Embryology Lab tonight. I want to just basically show you, what we do and why we do it. And I'll be touching on a little bit of the scientific logic behind everything that we do. My plan is to not go into too much depth with the science side of things because I am aware that it is getting on in the evening and I don't want anybody to nod off if it turns into a biochemistry lesson. So first slide, I've got a picture here on the right. This is our embryology lab at City Fertility. It's not a huge room at all, but it's got some really important equipment in it. That equipment is there to basically provide optimal environments for egg, sperm, and embryo culture. We try to keep the conditions as close to within the uterus as possible, as far as we know. We also have strict measures to eliminate external contaminants, so no infections, nothing like that. And also the environment within the lab is always constantly monitored to make sure that, should anything go down, any incubators start to malfunction, the embryology team are notified instantly, and we basically get there as quickly as possible to minimize any damage that could happen. Okay, so our logic behind what we do in the lab. So we try to think about what's happening in the uterus. At the end of the day, the best environment for an embryo is in a healthy uterus. So that's kind of our model that we start with, and we work from what we know about that. So we've got things like a neutral pH, nutrients available, nice cozy body temperature. Obviously, there's no light in the uterus, but most importantly, no UV light. No germs, no contaminants, appropriate water content, and also reduced oxygen concentration. So I'll go through these in a bit more depth now. So we'll start off with nutrients. Obviously, embryos, eggs, sperm need available nutrients in order to function, to grow. The nutrients that we provide to the embryos and eggs and sperm comes from the culture media that we prepare them in. So different culture media from different manufacturers will have different energy substrates in. Mostly though, we have glucose, lactate and pyruvate in the culture media that we use. These are used at different stages throughout an embryo's development. So pyruvate tends to be used all the way throughout. Lactate can be used in the first few days, and glucose uptake is significantly increased as we approach blastocyst stage. So that's at around day 4 to 5. Within the culture media, we've also got amino acids. These are very important in terms of building blocks for protein development, also help to build DNA. They also have a lot of other roles which are still being explored to this day. We don't know exactly all of them. We know that they benefit them. The exact concentration and the actual amino acids that are in each culture media, it's not easy for us to say particularly because the manufacturers don't necessarily publish that as freely as we would like. Obviously, it's a little bit of a secret recipe kind of culture going on. So we know what works and we know what we need, and we also just know that the culture media provides it. So aside from nutrition, we've obviously got oxygen. So all of our cells need oxygen to live. Now, the interesting part is that embryos don't need the same level of oxygen as we do as fully grown humans. We obviously live in around 20–21% oxygen in the atmosphere. But what we find is that embryos actually develop better at around about 5 to 6% oxygen levels. So this is thought that the uterus will obviously have a lower concentration of oxygen available to it. So after the mother is obviously absorbing oxygen into other cells as the blood's on its way around to the uterus, when it gets there, the partial pressure of the oxygen is slightly reduced. The way that we manage to reduce oxygen concentration in our incubators is to actually use nitrogen gas to displace the oxygen. So nitrogen is a very inert gas. It doesn't harm the embryos at all. But by pumping it into the incubators, it will push out the oxygen. So therefore reduce the concentration down to around about 5 to 6%. We also have water content. It's very important to maintain the water content of the culture media. So culture media tends to have quite a high content of water. I have heard estimates of over 99%, but as I've said, we can't be exactly sure of the levels of ingredients that are in there. We need to inhibit any evaporation of this water content while we're culturing embryos, because if we were to do that, then the concentration of any solutes and everything else in the culture media will increase. And by increasing those solutes, we will basically start to harm the embryos. So we inhibit this evaporation in two ways. With the culture media in the dishes, we actually put a thin layer of mineral oil over the top, and this will reduce any evaporation that can happen. Other incubators will also have a small tray of sterile water at the bottom, which we keep topped up at all times. This sterile water actually will evaporate in the heat of the incubator, and it creates a humid environment, which then reduces subsequent evaporation from the culture media in the dishes. So pH, I'll just touch on this because there's an awful lot that can be said about pH. And as I said, I don't want it to become a biochemistry lesson. But we do always try to maintain a neutral pH for the embryos to grow in. We use this with two types of buffer systems in the culture media. So the buffer systems basically prevent hydrogen ions from either being overabsorbed or overproduced. And it's the hydrogen ions that control the pH of the culture media. We can have bicarbonate buffer systems. These tend to be used for the longer term embryo culture, and they're used in most of our incubators. Anything that basically has CO2 being pumped into it will use a bicarbonate buffer system. So this buffer system is controlled by CO2 concentration, which is pumped into the incubator, as I've mentioned. And it does vary between labs, and it varies between different types of culture media as to which concentration is the best. The general concentration tends to be around 6%, but as I say, this can vary. This bicarbonate buffer system is used for the longer term embryo culture for the fact that as the living cells will metabolize, they will produce CO2. So anything that then produces CO2, we need to account for that, because the embryos can essentially increase the pH by accident by themselves if we don't account for it. So that's why we use the longer term bicarbonate buffer system in the longer term culture. The second buffer system is HEPs that we use. This is a bit of a complicated buffer system to explain. It's called a zwitterion. So if there are any biochemists out there, you'll probably know more about how to explain this than I do. But this buffer system is used when we have to take eggs and embryos out of the incubators and they are exposed to atmospheric conditions. So for things like egg collection, the ICSI procedure, when we strip the eggs of the cumulus cells, and for embryo biopsy, we will use this buffer system to maintain the optimal pH at all times, even though they're not in their optimal incubation conditions. And the most obvious one that we need to maintain is temperature. So we like to maintain egg, sperm, and embryos at a physiological temperature of 37 degrees C. We do this by obviously keeping the incubators at 37 degrees. Anytime that we have to take the embryos and eggs out of the incubators, though, they are always placed onto a heated stage. So what I've got here, this picture on the right, is our ICSI rig, and you can see this square here in the middle. This is the heated stage, which is heated to the right temperature to keep any dish on top of that stage at 37 degrees C. There's the hole in the middle for the light to come through, so we can still use the microscope at the same time, but the dish is always maintained at that temperature. These are two other examples of heated stages that we will always use when we take eggs and embryos out of the incubator. So this white platform here is heated to 37, and then this black platform here is also 37 degrees C. Okay, nasty germs. As I mentioned, we don't want any infections in the uterus in order to create a successful pregnancy, and we also don't want them in the lab. Now, it's very important that we take a good number of steps to reduce any contaminants, any infections that might occur, because the ideal embryo culture environment is also the ideal bacterial culture environment. And if there was a bacterial infection, it can be devastating to embryo culture. So one of the things that we use is antibiotics in the culture media, and this is supplied by the manufacturer. All of culture media mostly will have antibiotics in it. It's usually gentamicin, but this can vary between different brands. We will also make sure that all consumables that we use, so any dishes, any pipettes, any needles, will always come in sterile packaging. It's always single use, and we will only ever open that packaging prior to using it, basically. So it won't be sat on the side, collecting any kind of particles that might land on it. Another potential source of contamination is, at the end of the day, embryologists. As much as we wouldn't like to admit it, we go in and out of the lab. We have things to do outside in the rest of the clinic. And so we need to make sure that we don't bring any of that contamination into the lab with us. So we will wear scrubs for lab work. We have hats, so our hair doesn't get in the way. Lab-specific shoes that are only worn in the lab, and we're very strict with hand hygiene, so always washing our hands as we go into the lab. We have a good airflow system in the lab, which helps to keep the air inside sterile, so no particles will go in, no airborne toxins, etc. So the filtered air is pumped into the lab at a higher pressure than the air outside of the lab. So this has the effect that only filtered air will go into the lab, and it will then push out into the other rooms surrounding the lab. We've got one-way vents on the sides of the labs, on the walls, that will allow the air to flow out. But should the air pressure in the surrounding rooms become higher than that within the lab, they will then close to prevent any air to flowing back in. The air is also constantly monitored for volatile organic compounds, or VOCs. And these are basically airborne toxins and chemicals that can have a detrimental effect on embryo development. So if anybody's watching who has ever had an embryo transfer and they've been told not to wear perfumes, for example, that's because the perfume will contain alcohol, and this is an example of a VOC, and it is toxic to embryos. So don't wear perfumes or strongly scented cosmetics, please. We will use class 2 safety cabinets when we are taking eggs and embryos out of the incubator. And this, again, helps to keep purely filtered air on the cells. And any contaminated air, any air from the lab, won't make it into contact with the cells. So the picture on the left is an example of one of the flow hoods that is in our lab. And there's if you can just about see, there's a glass screen that comes down around the eyepieces of the microscopes. And then the diagram on the right shows how the filtered air is pushed into the inner chamber of the cabinet. Any external laboratory air is then shut out of the inner cabinet, and any contaminated air is then sucked away and out of the exhaust, so always kept away from any cells, eggs, embryos that are inside. So Andrology Lab. So this is something that is important, and sorry if there are any men watching, but seminal fluid is not sterile. So we have to keep all of that well away from the eggs and embryos until the sperm has been processed and ready. So even though it's not sterile, that's not a bad thing. It just can contain normal bacteria that's present on somebody's skin. There might be cases where more harmful bacteria could be present. So what we need to do is always make sure that we essentially wash the sperm cells. We remove them from the seminal fluid, and then once they're prepared and clean, they can then be brought into the main lab just before insemination. It's important to know that eggs and embryos are never taken into the sperm lab. There's no reason for them to go ever in there, and it's just too high a risk of contamination. So we have some pictures here of our separate andrology lab. We have this separate flow hood here for the sperm samples to be processed in. And then over on the left, it's got its own centrifuge, its own microscope, and it's even got its own computer and desk and work area in there, so that we make sure that whoever is processing the sperm samples for that day doesn't need to be running back and forth between the main lab and the andrology lab. They can just stay there, process the samples, and then once they're finished for the day, they can come out. Okay, UV light. So obviously there's no light in the uterus, but most importantly, we need to exclude UV light from the embryology lab. So the reason for this is that DNA is denatured by UV light. So anybody who's ever sunbathed for too long and got a sunburn will know the dangers of UV light. And the same is applicable for eggs and embryos and sperm. So they don't have a protection mechanism against UV light like humans do. So if they're ever exposed to it, there's a high chance that their DNA will be denatured and then they won't develop. So we have UV filtered light coming from the ceilings, the microscopes. We tend not to have windows in an embryology lab. Any labs that do have windows in them, they will have UV filtered glass on them. You'll get some labs who, if they don't have UV filters in their ceiling lights, they'll have very dim lighting at least. So it's something that's very important, and we always try and control. Okay, so I'll show you some of the incubators that we use in our lab. This one in the picture on the right is quite a big incubator. We only use this to set up dishes and tubes of media for treatment for the next day. So by setup, I mean we want to equilibrate any media that is going to be coming into contact with eggs and embryos and sperm. We've got 6 doors on this internal glass door, so that if we do have to go into the incubator, it will hopefully minimize the change in temperature and the change in the atmosphere of the incubator. It's not possible to completely eliminate it when we have to take things out, but we minimize it as much as possible. We also have this incubator here, which has 10 individual compartments, and this tends to be used at the beginning or at the end of treatment. So any patient's eggs after egg collection and before insemination will be kept in its individual compartment, and any embryo just before transfer will be kept in one of these individual compartments. So as well as keeping the eggs and embryos separate from other patient samples, it's got quite a small volume within. And the benefit of that is that with such a small volume of gas in there, it makes it an awful lot quicker for the appropriate gas concentrations and the appropriate temperature to be resumed after we've gone in there to take a dish out or put a dish in. So with a smaller volume, it's much easier for it to recover. And then we have our Time-Lapse Incubator in the lab. So we have a brand new Embryoscope Plus, which is a lovely piece of equipment. Time-Lapse Incubation, you've probably heard about it. It is something that enables us to culture embryos from day 0 up to day 6 without ever having to take them out. So the benefit of this is that all of the culture conditions that I've just mentioned, so the temperature and the gas concentrations, these are all maintained perfectly throughout an embryo's culture. So obviously in the uterus, the embryo is not going to be exposed to colder conditions. It's not going to be exposed to high concentrations of oxygen. And we want to achieve that as well. And this incubator makes that possible. So if we look inside it, when we go to add a dish in or take a dish out, all we see is this very small compartment here. And this dish here is perfect for fitting in it. It just about fits in. And if you can see quite closely, we've got 16 individual wells here, and that's what each embryo will sit in. So as you can see, when we go to put a dish in or a dish out, at no point is any other dish exposed to the external environment. So it's a really, really nice piece of equipment in terms of maintaining that steady culture environment for the best optimal growth. And so from time-lapse incubation, you can see these lovely pictures that we get. So with the Embryoscope Plus, I think it takes pictures every 10 or 20 minutes. And that means we can see everything that an embryo does without ever removing it from its optimal environment. And we can see so much more that we could never see before when we were having to take them out at certain time points throughout the day. So whereas in the past we would have maybe seen an embryo 5 times in its development, now we can see it for its entire development. So as well as maintaining the optimal environment to give it its best growth, we can also see everything that it's doing, and it gives us the ability to select the best embryo from what we've seen. Okay, so another important thing to consider in the embryology lab is security. Now, obviously, if you have an embryo in your uterus, you know that that's yours, you know how it got there. So in the lab, we want to provide that exact same kind of security as well. We have 2 systems that we're just introducing into the lab at the moment. One is called RI Witness, and this is where you have a heated stage which reads electronic tags that are put onto each dish. Each electronic tag on each dish is assigned to that exact patient. And any dish that then gets assigned while that dish is on the stage has to go to that exact same patient. If there was ever an accidental mismatch, so 2 dishes were put on that were assigned to 2 different patients, a big alarm goes off, red light flashes, and the embryologist is notified that something is not right immediately. We also have the Matcha barcode reader, and these barcode readers are positioned by the microscopes, by the heated stages, and the dishes are labelled with the barcodes and the patient details. The barcode readers will obviously detect the barcode. They also take a picture of each dish, and any time that another barcode is put next to this dish, it must be for the same patient. Otherwise, the embryologist is notified immediately again. So here's a little example. If you can see here, we've got this barcode reader here and another barcode reader here, just overlooking the heated stage, making sure that everything that's put on it is correct. And this white stage here, this is the RI witness stage. So any dish that is put onto any part of this stage will always be registered as belonging to a certain patient. Okay, so now we move to a very different kind of environment, and that is the cryogenic storage environment. So this is the room that we keep all of our frozen samples in. So eggs, sperm, and embryos are kept in each of these tanks here. This is a separate room from the main lab, and it must be separate for the embryologist security now, because liquid nitrogen, if anybody has ever worked with it, you'll know it's very dangerous, and it is a bit of a suffocation hazard if something should go wrong. So this room is kept entirely separate, and it has an incredibly good ventilation system to make sure, should the worst happen, nobody is going to, suffer. So these tanks are monitored with remote alarm systems as well. So if anything were to go wrong, if any increase in temperature was detected, the embryologist would be notified immediately, and we would all come in and sort it out. So you can see there's the vapors coming off the liquid nitrogen. all the samples are stored at minus 196 degrees C. The samples, once they are stored in there, can be kept for very, very long periods of time. And depending on the method of freezing, they can actually be kept for over 50 years in some instances. So it's an incredibly different environment to what we grow the embryos in, but it's a very, very good environment for storing them. Okay, so that was everything I wanted to share with you about the environment of the Embryology Lab. I hope that it has been interesting for you. If you have any other questions, though, please do let me know.
Emily, thank you so much indeed for this incredible presentation. Lots of useful information indeed, and very, very interesting. Thank you so much, because I don't think we talk about it too much, and therefore definitely there are some questions, as you can see, because it's definitely interesting for the patients to know how it all works. And you have opened this kind of topics, because actually I've seen questions that are very, very similar to each other, and I will show you them right here. Okay, let's go ahead with the very first question first. So do you change media in a blastocyst culture when using a monitoring system like the Embryoscope?
Okay. Sometimes you can and sometimes you wouldn't. It will vary between labs, and it will vary based on which brand of media you're using. So you can get sequential culture media, which is the kind where you will have culture media that's optimal for embryos to develop from day 0 up to day 3, and then you would change it to a different type of culture media from day 3 to day 5. You can also get single-step culture media, in which it's designed to support embryo growth perfectly from day 0 up to day 5. This is what we currently use in the lab, because, as I've mentioned, with the time-lapse incubation that we use, the real benefit of it is that we never have to remove the dishes from the incubator until we get to the final day just before transfer. If we were to be taking the dishes out and refreshing the media, obviously that exposes the embryos to the conditions that we're trying to avoid. And there's also always a small amount of risk whenever you have to move a dish. So for our lab, it's not something that we do. We use the single-step media and we get good results from it. But every lab will vary based on what they like to use best and what they get the best results from.
Wonderful. Thank you so much, Bubble, for your very first question. Definitely interesting one. And of course, Emily, thank you for answering that one. Okay, and this is now the question. And actually, there are some questions that are very similar to this one. Let's have a look. So why should no makeup be worn during embryo transfer? And let me just go to the next one. So is it recommended not to use skin cream or makeup during retrieval or transfer?
I see. Okay. So no makeup in transfer. I have not advised patients not to wear makeup during transfer. I know you don't wear it during egg collection, particularly if you're having sedation, because there are signs that the anaesthetist will look out for on your face, which makeup can hide. Makeup worn during embryo transfers, I don't know if that is a particular problem unless there are certain VOCs in it. So if there's alcohol in it, high alcohol content should be avoided. But if you check on your makeup, if you check on the ingredients list, and if alcohol either isn't in there or it's quite far down the ingredients list, it's probably safe to use. But if you're worried, if you don't want to take the risk, then just don't do it. It really is up to you. But I haven't advised people not to wear makeup during a transfer. And then was there the second one about skin cream as well?
Yes, exactly.
I think skin cream is fine. Again, as long as it's not heavily scented and doesn't have a high content of alcohol in it, then it should be fine. You know, I will use skin cream on my face daily, not scented, obviously, but, you know, I think I would dry up if I didn't. So I do use.
Okay, wonderful. Thank you so much for the clarification. One more, okay? It's similar, but is using a skin cream with perfume harmful during egg retrieval or embryo transfer?
I think it is dependent on how scented it is. So I would advise you to use non-scented skin cream. It's all about the alcohol content, though. I would always recommend you avoid the alcohol content with anything. Okay.
Excellent. Thank you so much indeed. Definitely interesting, because I don't think everyone would think about it even, at some point, if it wasn't mentioned. So indeed, this is definitely interesting. Thank you. All right, let's have a look at the next question. What is the state of in vitro egg maturation in a lab? I have some M1 and M2 eggs frozen. Could I expect my M1 egg to survive the thawing, get mature in vitro, and fertilize?
Okay, so our lab doesn't do in vitro egg maturation. It's something I don't know if it's widely done in the UK, and that might just be because of the HFEA and their restrictions and things. With M1 eggs that are frozen, it is possible that it will survive the thaw. There's no reason to think it wouldn't. And again, hopefully it can mature. It will depend on when it was frozen and how long it was left to see if it would mature before the freeze. Now, what I would probably advise if it matures the day after it's thawed, for example, so it does mature, it does fertilize, and if it creates a blastocyst, I would probably advise maybe having a genetic test on it just to make sure that there's not aneuploidy there, because there has been research that shows that eggs that mature quite late might have quite a high incidence of aneuploidy. So basically, where the chromosomes aren't balanced, and it could lead to other problems. So if it fertilizes, if it gets to blastocyst, you might want to consider doing some extra tests on it. There's no reason for me to think that it wouldn't survive the thaw, though. Everything else is quite dependent on the actual quality of the egg.
Again, thank you so much indeed for yet another interesting question, and of course for your help with that. Again, we will go to the previous questions on perfume, but let's have a look. Okay, it's definitely
scared everybody. I'm sorry, I didn't mean to scare anybody.
It's good. It's good that you did, actually. This is definitely interesting. So is it a problem the patient had used perfume before they transferred that?
I think if you've had your transfer and you've used perfume, don't panic over it. It's something that we do recommend that people avoid. And maybe if you have transfers again in future, don't wear it. But if it's happened before, if you've already had your transfer, don't stress, because there's a lot of things that we need to consider, and a lot of it is all about just reducing risk factors. So yeah, we advise against perfume. I would not recommend it for any future transfers, but it doesn't mean that that's the end of it. It's not that powerful that it could have ruined anything. It's just something that we would say, maybe do it just to sort of increase your chances ever so slightly.
Perfect. Thank you so much for the clarification. As you can see, lots of questions on that. So this is something that I guess no one really has expected as well. So thank you so much for mentioning that in that case, of course.
No worries.
I'm typing, okay? I just need a minute to see whether we have another question or not. But of course, I'm sure, as you can see, it's very interesting. So if you have any left, go ahead and do it now. We would love to see what else you would like to ask, that's for sure. And we just need to, yes, there it is, sorry.
Yeah.
Let me show you. So are blastocysts on day 6 or 7 not of good quality? Sorry.
Okay, day 6 blastocysts are fine. I wouldn't worry about a day 6 blastocyst. We do see good results from transferring day 6 blastocysts in relation to day 5. Day 7 blastocysts, we don't culture to day 7. It's not something that's routinely done, I think, in the UK. It has been done, but again, I think I would probably recommend maybe a genetic test if you have that available to you for any day 7 blastocysts. It's not something that we tend to sort of wait to see if it happens. We tend to have day 6 as the cutoff, but again, I wouldn't write them off. If you have them, if you've got good quality day 7 blastocysts frozen, but I would probably say take extra precautions before having them transferred.
And wonderful again. Thank you for the clarification. Next question is right here as well. What are your thoughts on genetic testing of embryos for aneuploidy? Is it reliable?
Yes, yeah, I would say it's reliable. With everything that we do, it's not 100% perfect, but I would say that if you have a good reason for wanting your embryos to be genetically tested, so for things like recurrent miscarriages, or if the female partner is maybe a bit of an increased age, then yes, it can be worthwhile, definitely. Obviously, when we're selecting embryos in the lab, we will watch how they develop. We watch the quality of them on day 5 or day 6, but we can never tell what's actually going on inside the cells. And if you have increased risk factors for aneuploidy, or something that could, change the chromosomal makeup of the embryos, I would say it's definitely worthwhile to at least consider it. Have an appointment with a genetic counselor so that you know what you're getting into. And then if you think that it's something that would work for you, I would say it's a good option to at least give you peace of mind that any embryo that you're having transferred back is chromosomally normal. It's just worthwhile finding out if you could be in one of those risk categories.
Fantastic. Once again, many thanks for that. Okay, let's have a look. Okay, we do have another one here. In Germany, testing of embryos is not usual or for all. Okay, are there other ways to get to know the quality? Egg testing is it possible?
So egg testing, it is possible. So you can take a small biopsy of what's called the polar body from the egg. It's not something that I think is particularly reliable. We don't, I'm not sure if we do it in the UK still. The reason being is that you can get the quality of the egg. So maybe it could help in terms of ruling out a particular embryo, but you can't guarantee that any embryo that has developed from a perfectly healthy egg is still perfectly healthy. So it's something that is not as reliable as testing the embryos, on day 5 or day 6. Other ways to get to know the quality, they are developing ways that we can get to know the quality without doing a biopsy of an embryo. So they're hoping to develop ways of testing the culture media. So what the embryo has grown in, if you can actually test that media, you might be able to find out if there are chromosomal abnormalities in that embryo. It's not perfect yet though, so it might be a way off coming, but there is something on the horizon at least. And then it's just things like if you have access to time-lapse incubation, that can really help, because if you can see the divisions that the embryo has made, it can definitely give you a good idea of, is everything going on as you would expect in that embryo. It's not as perfect as actually doing the genetic testing, but I understand it's not available all the time. So it could at least be something that could help.
All right. Again, thank you so much for that. Margie has added one more. Good news. When is it coming then? Anything you can
Oh,
add?
the culture media testing. Oh, soon, I hope. I really do hope soon. I couldn't tell you. Unfortunately, I'm not involved in developing it, but I know there are a lot of companies that are looking into it and perfecting it. But obviously, they need to make sure that it is reliable. So it's something that they are testing, they are validating, but it could be years. I wouldn't be able to tell you how long it's going to take, I'm afraid.
This is again one of those things we just need to wait and see. Yeah, exactly. Thank you so much, though. Of course, definitely interesting. All right. Next question. What do you think about add-ons such as assisting hatching, etc.? Are they really useful? Do you use them?
Yeah, add-ons have had such a bad press recently. Some of them probably rightfully so, and some of them, they're just getting a bad time because the gold standard of scientific trials, so a randomized controlled test, just can't be performed on it. So things like assisted hatching can be very useful. So if you're in the sort of risk category, so if you've had repeated failed implantation, for example, if you are having transfers of frozen embryos, and again, if you're in an advanced age category, sometimes, yes, it can be really useful to have something like assisted hatching. And with assisted hatching, it's such a simple process. And when it's done correctly, it doesn't damage the embryos. And yes, at least you know then that the embryos will be able to hatch out of their shell, of the zona. It's difficult for me to say, yes, they definitely work, because, all the scientists, all the embryologists in the country can't come up with scientific proof that happens. But it's something that we do in the lab. We do believe that there is good reasoning behind it. And if somebody has had the repeated failed cycles, then, why not try something different that won't harm the embryos? It won't decrease your chances, and it might just increase your chances. So it's something that I think is worthwhile.
And once again, thank you so much. A little bit of topic here, but I'm definitely interested for all the patients. I definitely love the questions that you are putting here tonight. Okay, let's have a look. Okay, definitely interesting. Here, another one. Do you use IMSI and PICSI? And if yes, for which indications?
Okay, so we use PICSI more routinely than we use IMSI. So PICSI is basically where you do ICSI and you use a dish which allows the sperm, or only mature sperm, to bind to a hyaluronan which is on the dish. So this is something that is found in the shell of the egg. So the theory behind this is that any mature sperm which would have the physical capability of fertilizing an egg will be bound to the bottom of the dish, and we then go along and select the best looking one, basically. Anything that's not bound to the dish, we know it's not mature. So it gives us an extra level of knowing which sperm to pick while we're doing an ICSI. So indications for that, I think if you have had a sperm test which has suggested that you have a higher concentration of immature sperm, so sperm that doesn't bind to hyaluronan, then yes, they might suggest PICSI, just because if you've got the higher concentration, there is a higher chance of an embryologist selecting an immature sperm by accident, because otherwise without it we can't necessarily tell. IMSI is basically doing ICSI but with a higher magnification. So if there are things like vacuoles in the sperm head, or if the sperm tends to be misshapen, then we might use IMSI just to make sure that we are looking as hard as we possibly can at each sperm cell that we're selecting. Again, it depends on the semen analysis that you might have had, if it's recommended for use.
All right. Thank you so much again for the explanation. And another interesting question. Okay, Elle has added one more question. When selecting a clinic and lab, what should I ask in regards to embryologists and their experience, etc.?
Ooh, that's difficult now. I suppose you'd want to know things mostly about success rates, really. I mean, you can go into all of the details. You could ask for, all of their service reports, things like that, all of their qualifications. But at the end of the day, that doesn't mean that they're going to be doing, the best work for you. I think if you can look at the success rates, the individual success rates of a clinic, that's going to give you a much better picture, because at the end of the day, it's not just the embryologists that will contribute to your treatment. All of the nurses, the doctors, the stimulation that they give you, the procedures that they do, it's so important to take all of that into account. So I probably wouldn't advise you to only look at the embryologists when you're selecting a clinic, but look at the success rates as a whole. And if you can, look at the treatments that they offer in terms of, IMSI or PICSI and genetic testing, if you can. Anything that you think you might want to sort of look into, just because then at least you have the option if you need to without having to change clinics. But yeah, I wouldn't necessarily know what experience you should ask for. I think success rates will probably be the better measure of how good a clinic is.
All right. Thank you so much again. Next question is up. Are the results with ICSI and IVF the same if the sperm is not an issue?
Yeah, that does. It will vary between clinics, but yes, generally speaking, ICSI and IVF have comparable success rates. As I say, it's going to vary between clinics, and I can't comment on everyone's success rates, but certainly in the clinics I've worked at, ICSI and IVF do tend to be very comparable.
Excellent. Thank you for the clarification. Next question. Which ingredients do you use in your culture media?
I have no idea. And as incompetent as that probably makes me sound, the reality is we know that our culture media works. We validate it for the processes that we do. But in terms of the ingredients, that's not something that the manufacturers actually publish to us. So we will know that they've got gentamicin in for the antibiotics. We know they've got glucose and pyruvate in. Much more than that, they don't tend to share with us. So it's very difficult for me to answer that one, I'm afraid.
Well, in such case, thanks for the honest answer, of course. Okay, thank you so much indeed. Okay, let's have a look. Margie has added one more question. That might be our final question. So if you have any left, you know what to do. Okay, and Emily, let's have a look. What are the survival rate of egg and also embryos after thawing, I guess, yeah, in your lab?
We have good rates. So embryo thawing, I think we're at about 90 to 95% survival rate, which is good. Egg survival rate is always slightly lower, just because of the nature of egg cells and how they survive. I'm not sure of the exact survival rates of eggs after thawing. It's probably, well, it's usually around about 60 to 70%, I'd say.
Wonderful. Thank you so much for the clarification. Well, someone is typing, so yeah, here is the next question. What is the temperature?
In the lab, it's a nice, comfy, about 23 degrees generally. So we don't obviously keep the lab at 37 degrees because we'd all pass out. But yeah, we keep it at a nice warm temperature to minimize any effects on taking any dishes out.
There's just one comment here after you answer, so exactly. Thank you so much indeed. But it's true, right? So thank you so much for. I actually, of course, there are some thank yous and thanks coming up your way because, of course, we will be finishing. But it's been an incredible session, that's for sure. And for some of you probably don't know, but this is actually Emily's first webinar. I wouldn't guess, really. So thank you so much indeed, Emily. It's been a brilliant, brilliant session. And someone has a comment here. And really, what can I say? This is what I would like to say to you. Thanks so much, Emily, for taking us into your lab.
You're welcome.
That's definitely something that I would agree on. Thank you so much because it's been a great session. One more question is here in the meantime. So which pH do you use?
So our pH tends to be around about 7.2. So that's, again, it will vary between labs, but 7.2 is what we calibrate our media to.
Wonderful. Thank you so much. So one more thank you for you. And of course, we will be finishing. However, before I let you go, Emily, anything else you would like to add?
Oh, I honestly don't know. I just hope it was interesting and I hope I helped with some of the questions at the end there.
You definitely did help, I am sure, and it's been a very interesting session and lots of questions for you. So I'm sure it's been useful, and you've been, as I mentioned, you've been brilliant. So what I can say Let's just do it sometime. Okay. Again, there are so many topics we could discuss for sure. So thank you so much indeed for your time, for joining us tonight, and everyone for your incredible questions. As always, you never disappoint. So thank you so much for coming in here and asking, because if you are asking, that means that what we do is helpful. We are happy about that. And well, Emily, thank you so much. And I just want to mention that this has been recorded, so if anyone missed any parts of this, remember, you will be able to join my IVF, of course, on our website. It's out there. There are over 300 webinars. You can check it. There are other topics out there available. But I can also encourage you to simply sign up, because if you don't know already, we will be back here tomorrow at 8 p.m. UK time. So I hope you will be able to join us. And of course, Emily, again, thank you so much. It's been a great session. So thank you so much. Have a lovely evening, everyone. And till our next event, okay, Emily?
Yeah, definitely. I'll be there.
I'm glad to hear that then. Thank you so much. Take care, everyone. And I hope to see you back here tomorrow. Take care.